make some comparisons for what we saw with this crab to what we do. Okay, so for example, boys, y'all may not know this, but this is a comb, and it's called it's called a wet comb. Okay, and what it is that girls usually are going to use this on hair that's wet and really tangled, and it helps detangle your hair. Okay, and it works really efficiently at detangling your hair. Y'all, this just looks like serrate CD to me. It has the long shaft, and it has these teeth right here, right? So my hypothesis is that serrate CD are for detangling, maybe detangling the lamellae of the gill structures, detangling things. Now, again, boys, I don't expect y'all to know that this is a round brush, okay? What you can see from this round brush does not penetrate the hair. It doesn't detangle, okay? But what we know, girls, that a round brush does is it does fine detail to your hair. It helps make your hair straight, okay, versus like curly. And so I'm kind of hypothesizing this is what Pappos CD does. Pappos CD is too fine. The sexuals, little pointy things, are too fragile to actually go in and detangle. But what I'm hypothesizing is that Pappos CD does some like fine detailed kind of removing fouling. And so we found a ton of different CD on all the grooming appendages. Some interesting things. That weird cuspidate was only found on the feeding appendage. Type 4, only found on the feeding appendage, okay? So I'm guessing that these guys are only kind of have something associated with feeding, which is so not my specialty, okay? When I look at what's going on in the gills, serrate type 2 and type 3 were only found on the epipods. So they're associated with cleaning the gills, probably detangling the gills. So the gills have increased surface area for water flow. So this is your last one. So spider crabs have different types of CD used in grooming. That's pretty easy. Your chemistry test was this easy? No?
Some of them haven't slept in days.
That's great. Well, y'all are not sleeping right now, which is a good thing for me. Okay, guys, so I'm going to accept that hypothesis, okay? They have very elaborate cedar morphology, okay? It's not necessarily what I thought it was going to be, but they do. They have different types of serrate CD that is used for detangling of structures. They may have some that represent that round brush like Pappos that are for fine detail cleaning. And then we know that we found 8 total types of CD. 2 were just associated with feeding, and then 2 were just seen in the gill chamber, right? So kind of to the big picture take home with this, okay, is that first of all, they decorate not to match, but they decorate for disruptive coloration to not look like a crab. The second thing is that larger crabs did not decorate more than smaller crabs like I thought they would. Larger crabs don't groom more than smaller crabs. Thus, larger crabs may have a different strategy for concealing themselves from predators. So this is kind of an interesting hypothesis, guys, that has not been presented before, is that smaller adult crabs have different strategies for concealment than larger crabs do, right? Which is something that's totally something that we haven't really thought of before. We know that spider crabs are not grooming the regions where their decorations take place. So they're just basically saying, okay, I'm going to have these CD until I molt, and I'm going to get brand new ones, right? And then they have grooming behaviors, I think we're all agreed, that are secondary. They only occur if primary behaviors are not present. That spider crabs behave like other decapods and that they're grooming sensory and respiratory structures, and that their grooming time budget is much, 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 much less than other decapods, okay, specifically shrimp. And that's even with them having different structures than shrimp, these decorating CD, and having their gills in an enclosed chamber, which shrimp don't have. So these crabs are not grooming themselves very much. And these spider crabs do have different cedar morphology from other decapods. A couple types of serrate CD have not been found in decapods so far, and that they have really good cedar morphology for grooming their gill chamber. So where am I going from this? This summer I'm going to be actually, guys, looking at specifically under the SEM exactly where the decorating CD occur. I'm going to be looking at differences in the types of CD on the carapace region between larger and smaller crabs to actually see, do larger crabs have decorating CD? I'm predicting that they don't. but no one's ever looked at that, and also to look at if there's differences between boys and girls. And so with that, that's my talk. Thank you for staying awake. This is a female that's actually grooming her eggs, which is really cool. Are y'all have any questions? Yeah.
Okay, so no offense is meant by the way I'm going to word this, but
I have no
you
fear.
got us there. But so is it possible that decorating crabs are like the hippies of the crab world? So I kind of like, right, they're putting stuff on them. They might not want to like clean themselves off.
It's possible that in order to conceal themselves, right? So if you think about it, if I have CD attached, if I have like algae attached to me, I might not be able to get to my decorating CD, right? It is possible, but then that gets us to the problem of these larger crabs that don't have decorating CD, like they're going to reach a terminal. If they don't groom these CD, they're going to just wear off in the environment. So I would predict, I agree with you that when crabs are smaller, maybe they don't want to decorate, maybe they don't want to groom themselves. But I would think that larger crabs, from a natural selection standpoint, I would want to groom those decorating CD so I can decorate as long as I can to have a predatory concealment strategy that's worked really well. So that's what I would predict. But I agree with you. They're not grooming at all like shrimp. It was really startling to me, actually, to be honest with you. And it makes me worry about like blue crabs and stone crabs. So that's kind of the next thing that I want to do, is that I want to kind of replicate the study, but I want to do it on blue crabs. And I want to look at how blue crabs, if blue crabs also, so I have a different species, a different sample size, to say, okay, do blue crabs really groom at like this ridiculously low 5% or less budget of time grooming? Yeah.
Do other species like blue crabs and stone crabs have these?
They don't. They don't. So that's exactly right. They don't. That's also why I want to do it. So I would predict that, first of all, they're not grooming their CD. It's really not happening. Is their time budget like of a blue crab and stone crab probably going to be less? Absolutely. Which is frightening. Sean?
Would you expect that the amount of grooming to occur in the tank experiment to be less because it's nice, clear water?
Well, that's great, because my water quality that I use is that are my animals in clean water from like a chemical standpoint, like the amount of calcium and everything in the water? Yes. But my water, I'm not one of these people like that has the tank out here. My water is in, I think it's an 800-gallon recycling thing. I don't change the water. I don't change the filter. I don't keep it pristine clean. Do I have algae growth in that water? Absolutely. And so my environmental conditions in the lab, are they probably better than what's out near Fort DeSoto when they're buried in the sediment? Probably a little bit. But my water conditions were not pristine, and I do that kind of intentionally.
Other questions?
Y'all have like all these weird sailboat things going on. I just kept seeing shit flying by. Sorry for the cussing, but I was like, what is going on out there?
Come up to my office. I can't get anything done.
It's like if you have like attention deficit disorder, it's like, oh my gosh. Are y'all good? Thank you guys for staying. I'm sorry if I kept you long.